The topic multi-line breeding, and we also discuss different types of breeding, multi-line breeding and mutation breeding if time permits. I will just finish my presentation and everyone can join. So, the multi-line breeding, actually the term was given by the Jensen in 1952, and he defined that multi-line breeding it refers to mixture of near isogenic lines or resistant to a specific race of a pathogen, or it's a deliberate combination of isolines, or it may be a closely related lines, or maybe it consists of unrelated lines also. Entirely, we can pool them. It is called as a multiline. How we can develop a multiline? Either we can develop first you have to develop the component lines, then you have to screen the component lines, then you have to group the component lines, then you have to go for seed multiplication program. Then how to develop the component lines? Actually, as earlier defined, multiline is a mixture of pure lines. It means that you have to develop pure lines first. Then it should be isogenic, or it may be closely related or unrelated lines. But when you take the population as a whole, they should have equal maturity and equal agronomic performance, and they can only differ for the some resistant genes or some desirable genes. But most of the agronomic character should be similar, even though they are unrelated lines or related lines. Either it may be isogenic lines. Isogenic lines, there will be no problem of agronomic characters. But when you are using related lines and unrelated lines, they should take care of the agronomic character should be similar, and they should matures at the same time, and they should give the yield should be same as like other individuals. This is how you have to take care. Like when you're grouping the lines, or when you are selecting the lines, you should take care of all these parameters, agronomic characters and quality parameters, all those things. And you should be homogeneous, but they are heterogeneous, because we are also considering a group of unrelated lines also. Generally, we can say that multi-lines may be the heterogeneous topics. Then, usually, how many lines we can able to use is up to 16 to 15. More will be the number actually good, but there will be a limit. After the 15, what will happen? Maintaining it will be a very difficult task, because 15 lines means the population will become very large. Then you can't maintain that population. That will be the another difficulty. And too less is also a problem. That's optimum like 6 to 15 lines should maintain. Then if it is isogenic lines, and it will take a lot of time to develop. We have discussed how to develop isogenic lines. Normally you can develop isogenic lines so that like every line should have all the resistant genes, or each lines have a single resistant genes for the it based on the need, like how many rays present in the particular population. These are the things we need to take care. Then normally how to develop isogenic lines, we need a 5 to 6 back crosses, and you can go with any system of mating. Either you can go either parallel back crosses, stepwise back crosses, or multiple back crosses single time. That is the it's depend on time and ease of the crossing. Then what is the genetic basis of these multiline varieties? As when Van der Planck has discussed, you know that why we need to develop a multilines, because in a multiline we can able to utilize both vertical resistance and non-specific resistance, like called as horizontal resistance. Like xt is equal to x naught e to exponential to the rt, where rt is your horizontal resistance. Means when you are including a different diverse line into the population, then definitely when you look at the sum total of the all the population, it looks like it mimics the nature of horizontal resistance. But when you consider individual line, like every gene may having like specific to the particular race or a particular race of the pest. You are taking the advantage of both vertical resistance and the horizontal resistance, so that we are delaying the particular race, the epidemics of the particular race or a growing of the particular race in the particular area. This is the strategy that is used by the Jensen, and later the Borlaug also used this strategy to develop different multilines in the wheat. Then, what are the advantages of these multilines? One is you can able to synthesize it mimics like a horizontal resistance. It can also called as synthetic horizontal resistance. Then prolonged life of resistance genes, because we are using a multiple resistance sources or genes at a single time, so that the pathogen developing a resistance for one particular origin, it will be it will be easy. But when you're having multiple genes, it will be very difficult to develop resistance against all the genes at a single time. It's very very difficult. That's why there will be the durability of the particular resistant genes will increases. Then the longer life of the varieties, as because of the durability of the resistance is increases, then definitely we can use the varieties for a longer time until the super race or any race can overcome all the resistant genes. Okay, then linkage and allelism for disease resistance at particular locus can be easily increased in a single variety. Like we have discussed, what is allelism, what is linkage means. For example, you take, there will be a specific R gene for a one race of pathogen. Due to we are searching for the uh, s- different alleles for the particular gene for that for that particular pest, so that we can increase the multiple alleles for the particular gene, so that we have a broader genetic base for a particular for a particular pest. This is one of the striking advantage of the multi-lines over the commercial other conventional lines. But what are the limitations of these multi-lines? It will be very expensive to develop and agronomically conservative. Why? Because you need to develop isogenic lines or related lines. Then it will take 5 to 7 years, and it will, the doing back crossing is a very very difficult task. At every generation, you have to do back crossing. It will be a very tedious and it requires more labor and it will be expensive. Then agronomically conservative. As I first initially I discussed, all the lines should have a similar agronomic character. Ultimate aim of the breeder is yield. You should not compromise with the yield. When you are developing a multiple source, like when you are using a multiple lines, then definitely you should take care of the yield as a first objective. Then if your goal is in yield, then you should not compromise with all your agronomic characters. And also for the mechanization also, you should be take care of uniform maturity, uniform time of flowering. Then everything you should be take care, you should taken care. That's why doing that it's very very difficult. That's why it's agronomically conservative in nature, that developing the all the lines which are having a similar agronomic character at similar stage of maturity be very very difficult. That is one of the limitation of multilines. Nowadays nobody using multilines because of these limitations. Then if possible, I told like developing of resistance against R gene in the particular pest, it is when you're using a single monogenic gene, it will be very easy to develop a resistance. But when you're using multiple genes, it is the development of the resistance particular R genes in a pest, it will be delayed. But unfortunately, if pest can develop a resistance for all the resistant genes, then it will become a super race. Then you have to search for a new gene, because you already used all the particular resistant genes for a particular races of the particular area. But all the races have overcome the resistance of this gene, overcome the resistant effect of all the resistant genes. Then from where you will bring the new resistant genes? Then it will need a lot of time, and it is a lot of time and lot of search. You have to do a lot of efforts to search the new genes, and it will be very difficult to bring that into the breeding population. Then again developing the lines to develop particular super race, it's very very tedious task. And there are several example of development of super race in several crops. That's why, but this is very very rare actually, because when you look at the genetics, developing the same time and having a resistance, overcoming the resistance of the all the resistance gene at the same time, it is very very, the chances is very very less. Okay, then these are the limitation of multilines. What is the different example of the multilines is like Mirama 62, like it is developed for wheat stripe and stem rest resistance, and different MLKS and 11 KML to 2404, KSML3, and Sentica multiline 1. These are several examples in medicine also it mentioned. Please read it. Then what are the different types of multilines present? Either you can develop multilines by mixture of several isogenic lines. Then either you can develop the multilines by mixing the closely related lines, or you can use unrelated different genotypes to develop the multilines. Then there are the two different approaches called as clean approach and what approach means every component all resistant genes, all the lines. And is very clean for the from your inoculations. No, no all rays, all relevant in that area cannot able to infect particular lines because it consisting of the all the resistant genes in a single variety, and every line consists of all the resistant genes. That's why it is called as clean crop approach. But as a layers, the super rays in this case is very to overcome this. The another approach, directly crop approach, not if you don't keep all the eggs in a single basket, like they were saying, you can't keep all the eggs in a single basket. The same approach here also followed means every line having a some resistant genes, but it's not having all the resistant genes. Having some resistant genes, some vertical resistant genes, but not all the resistant genes. But all the lines, when you take the sum total, it having all the resistant genes for a particular races present in a particular area, but not in the single component. But earlier clean crop approach, we were stacking all the different resistant gene into a like all single lines. Then you are develop every line of your multi lines are consisting of all the resistant genes. But in the tritic cup, it having some resistant genes, but not all. But when you take multi-line population as a whole, it having all the resistant gene for a relevant traces of the particular area. This is called as dirty crop approach. Here, development of super race is the chance is very less. Then this is about multi-line breeding. It's a very simple concept. Like what you should remember what is multi-line, what are the different steps to develop the multi-line. Then uh, what is the genetic basis of multi-line variety to take the advantage of both vertical resistance and horizontal resistance. Then what are the limitations? Then different examples. This much is more than enough for the exam. Okay. Then you should remember what is clean crop approach, dirty crop approach, who given it, Marshall has given it. Okay, then you should remember this much points is more than enough for the this particular concept. Then moving into the next concept called as clonal selection. Then, Miss, what is clone?
Excuse me,
It's a
sir.
yeah.
So both mean the same, no clean crop and dirty crop. Like every single line is having a single resistance gene.
A line having all resistance, if it is a clean crop, a line having all resistance. Like for example, you're having a 20 resistant genes, 20 different traces of the in the particular area. You to separate or stack all 20 genes into all the lines of your multilines, like all the population of your multiline. But in case of dirty crop, every line having some resistant genes, like one line having genes, another having another two resistant genes, another line having two, another two to three resistant lines. Every line not having all the vertical cover. In case of crop approach, every line having all the 20 resistant genes for a particular pest. All the particular pest. Are you understood?
Yes,
Not
sir.
the
Yes.
same. See, every line has a single vertical resistant gene, but in clean crop, it is not the single. Like all resistant genes incorporated all the length of matter. It's not strictly a single gene. Sorry. Yeah. Now my slides are visible. Yes. Hello. I'm audible. Hello. Hello.
Yes, sir, you're audible.
Yeah. Okay. Okay. Then there another breeding method is called as clonal selection. As you know that what is clone? It's a progeny of a single plant obtained by asexual reproduction. Then what is a clonal selection means? You are selecting a superior clone from the mixed population from the asexually propagating crop. It is called as clonal selection. Means you have a mixture of clones, some are undesirable, some having desirable character. You are doing a selection and you are isolating them, and by advancing to the like developing a population from out of that, then you are developing improved line. This is called as clonal breeding. Then what is the main important features of this clone is they're having homogeneous population as like self-pointed crop. They're having homogeneous constitution, but they're having some sort of heterogeneity. It depends on like crop to crop. Because if you take potato, having a homogeneous population, but when you look at the individual, because they are heterozygous in nature. But you take the carrot or radish, this one, there will be a highly cross-pollinated, and there will be some sort of heterogeneity in the population, and it's heterozygous in nature. They are having a very vigorous growth because they are asexually propagated, and they are heterozygous in nature. They have very vigorous growth, and they're having very wider adaptation because the like normally the asexual propagating crops are heterozygous in nature. They have the more recombinants can be produced. Like sorry, they're having generally a wider adaptation. Then they have very lot of source of like depends on like a source of variation like initial population because of this like most of the sexual crops are not that much selection was not happened during the course of evolution. Like take the brassica, there are a lot of diversity is present. You take the potato, there are a lot of diversity is present. Means there are a lot of variation present till now. That is why it's a like it's a one of the very important feature of the clonal selection. Then another important like a very important character of the clonal selection is they are having segregating population in F1 itself. This is many times asked in the exam. Like you take mango also. Like normally they're when you cross like F1, like they're actually pseudo F2 sometimes because when you are parents normally they are heterozygous in nature. When you cross, when you develop an F1, then definitely it looks like the pseudo F2. Okay, that's why they can segregate in F1 itself. It also called as pseudo F2. Then it is many times asked in the exam. Please remember, it is one of the important striking feature of clonal selection. Then why this source of variation occurs in this section? What by what means the like a source of variation occurs? Because it's not produces sexual reproduction many of the crops, but other source of variation is like either maybe bud mutations, like when it is propagated through bud, there will be chances of occurrence of mutations. That will be the one of the source for variation. Then mechanical admixture, like while you're doing transferring like asexually propagated propagules, then there'll be chance of mixing of two different like more or different varieties. Then by that means your variation occurs in the initial clonal population. Then some crops are amenable for sexual reproduction. That also one of the mean of the source of variation in clonal as like other crops. Then viral and bacterial infections, because once it is create any mutations, in generally in other crops due to continuous recombination through meiosis, that particular mutation may eliminate or may accumulate. But here in clonal crops, it's normally many of the times it is asexually reproducing. Once any mutations occurs, then it is fixed in the population. That's why, like viral and bacterial infection, if it is caused any mutation, it will be chances of accumulation of those mutations across the generations. It will be very high in these clonal crops rather than the other sexually produced crops, propagated crops. Then, what is the breeding procedure for clonal selection crops? First, you had to select the clonal initial population. It's a mixture of different clones. Then you have to select the best clone. Then after selecting, you just keep on asexually reproduce the particular generation to generation. Then because once you selected the best individual from that mixture, because it's a sexual reproduction, many of the times it is generally fixed in that generation only. But only thing you have to increase like population of that particular clone. Then you go for multi-location trial and release. This is one method. The another method is, you can, if you don't have any superior clones among them, you make controlled crosses among them, either through grafting or through any hybridization means. Then you develop a segregating population, because I said F1 only we can start selection, because F1 also many times as represent as a pseudo F2. Then you can do selection from F1 itself. Then you can fix. Once you select it, immediately fixed because of asexual propagation. Then you go for multi-location trial and release the particular clone. Then what are the important merits of this clonal selection is, it is used to for the conserving heterosis for the generations, because once it is fixed, because it's never going to recombine or never going to segregate, it is fixed. The heterosis what you are going to select in that particular population fixed across the generation. This is the one of the important objectives, like merits over the other different crops, like sexually propagated crops. Then they're like highly uniform, because there is no segregation and recombination many of the clonally propagated crops. Then it's the best method of isolating the best genotype from the mixed population. Then it can be immediately fixed also. But one of the demerits, like several demerits for this clonal propagated crops, like high prone to new rise of the pathogen, because I told, like, because once the some narrow genetic base happened means, once started infecting, because of there is no chance of natural recombinations, the plant cannot able to develop some evolutionary mechanisms, like a resistance mechanism for the particular pest, because it's not amenable for the meiosis, like segregation recombination. That is one of the problem. Maybe high, because of the epidemics of the pest will be very very high. Like you take the late beta potato, there are several examples you can see in the how the epidemics is causing devastating results in these clonally propagated crops. Then, as I said, clonal selection cannot create new variability. Then genetic makeup cannot be improved without hybridization. Like you have to make the control hybridization, then only you can create the variation. But normally it is a sexual propagated crop. It does not go for meiosis. That's why there is no segregation recombination. Then what are the problems while breeding clonal propagated crops or sexually propagated crops? Because there is no flower and very, very difficult for crossing. Because do hybridization, if there is no superior clones are available, then that will be a very, very tedious task. Then most of the sexually propagated crops, they are perennial life cycle. Like most of the tree crops, most of the fodder crops, it's very, very difficult to maintain the populations to having F6, F7 generation. It's a very very difficult, but we can fix at F1 only. But although to get the results, some like flowering fruit related characters, some flowering related characters, you need to wait a long number of time to select the particular trait. That's why perennial life cycle is one of the disadvantages to breed for these particular crops. Then difficulties in genetic analysis. If you have a number of, if you can able to take number of generations in a short duration type, we can have a pedigree history, so that we can trace the history of the particular traits. But if it is having long perennial cycle and asexual propagation, it is very difficult to study the inheritance of the particular trait. This is one of the problem while breeding for asexually propagated crops. What are the different achievements done by clonal selection is, there are several lines were developed, like varieties were developed, like Kurfi Red, Kurfi Sayed, Neelam Mango. Okay, Neelam Mango and CO series of sugarcane in the SBI Institute, and Kurfi Sundari. There are several varieties. And Kurfi normally it is in Shimla, it's a research station where the potato breeding taken in a larger scale. Okay, these are the different achievements as done by through clonal clonal breeding methods. You just remember the definition, and rest other things are same. And you should remember, you can select at F1 stage only. Many of the case, the F1 is itself is segregating. That one point you should remember. Then you should remember what is the problems actually. What's the problems in breeding for clonally propagated crop? It's a recurrently asked question. Means because of there is no availability of flowering, and most of these crops are like sterile in nature. Like self-incompatibility, male sterility are more predominant in those crops which are sexually propagated, and they have perennial life cycle, and they are tracing the history and inheritance of the particular trait is very very difficult in case of asexually propagated crops. Then this is about clonal breeding. Any doubts from this section of the topic? I'm going very fast because these are very very easy things. Any doubts? Then you can go for idiotype breeding. So just only remember definition, procedure, achievements, demerits. All the breeding methods. If you remember these four methods, no need to read again and again. Just remember the definition. Definition is very very important. Definition, procedure, achievements, demerits. These four things, if you remember from all the breeding methods, pedigree, bulk, single seed design, you should be in the tip like you should be very you should be in fingertips. Okay, whenever you like what is pure line, you should have like you should remember the definition. What is bulk? What is mass? What is pure line? Sorry, what is single seed design, and what is clone? What is clone? What is the clonal breeding? If you remember the definition, of the things will be done. And remember the unique character of each breeding method. In pure line method, progeny plant to progeny test, pedigree, keeping pedigree record itself is a, and the main striking difference between pedigree and bulk is in pedigree breeding method we can trace the history of or inheritance of the particular trait. In bulk, we can't do that because we don't keep any ancestral records. Then these points you should remember. Only these like very important points. And you should compare all breeding method. You should write in a single page. Like you should compare all the breeding method at a single go. What is the difference? What is the common things? And what is the demerits of each breeding method? If you do this one, it's more than enough for the exam from this all breeding from the section of breeding methods. Like if you do this thing means you can easily crack any question from these topics. That's why I'm very going very fast. Then what is idiotype breeding? And this is also very very important topic. And generally they will ask only definition and the procedure. Okay, and I'm not going to discuss with the types of video type, competitive video types. Okay, but just remember what is the definition of video type. But I'm focusing more on breeding by design today. Just remember what is idiotype, who given it, and the steps on the demerits. Okay, then idiotype breeding means in a broad sense like it's a biological model. It's expected to perform or behave in a predictable manner. Please remember while writing the definition also, these two words are three words very, very important. Like it's a biological model expected to, sorry, behave also I need to highlight, like should I highlight it? Biological model behave in a predictable manner. These three words are very important while defining the idiotype breeding. Means anything you just design on model like the particular plant to take rice, like last class we were discussing, you should have erect plant like erect leaves and semi-dwarf in nature and thick culm, moderate number of tillers, then high number of grains per panicle, high weight of the panicle. These are the several traits you are fixing. Means you are developing a model so that when you are breeding for that, you are bringing all the traits and you are putting in a single crop. They should behave in a predictable manner. Okay, you should yield. Okay, 10 kg means you should yield 10 kg. You should yield 10 quintal means you should yield 20 quintal in a particular defined environment. Means first you define the environment, then you should define the traits earlier only. Then try to bring the trait into the particular crop. Then expect that you should predict. You should behave in a manner that how you predicted the particular phenotype of the plant. Okay, in general, it is the only crux. Okay, then how you can develop this, we will discuss. Then Then first, what are the factors will affect while developing idiot big breeding? You should keep in mind which crop species you are doing like idiot breeding. Either it's a cross-pollinated crops, then which breeding method you have to choose. Then either it is self-pollinated crops, which one you have to choose. Either it is asexually propagated or the either non-sexually propagated. Is there any mesh systems are available or not? What is the percent of amenable for crossing? How do you maintain that population? Everything is character, which is the important economic traits. Everything you should take care. Like you should first you have to decide the trait means you should decide like how the botany of the particular crop. Then cultivation, like either, for example, you are developing a rice for DSR method, like direct seeded rice method, or upland ecosystem, or lowland ecosystem, whatever it may be. First, you have to the means of cultivation, like how they are cultivated. Because when you're defining the environment, then definitely the cultivation practices will have a greater impact on the behavior of the particular plant. Then you should take care of which cultivation method using for the particular plant in a particular environment. Then socio-economic condition of the farmer, either if you develop the radio type, like if you develop an hybrid, then a farmer can accept or not. Like you should remember, like then like these socio-economic condition of the farmer is very, very important while designing any crops. Then economic use. Like if you're developing basmati line, then you should take care of aroma, the length and breadth of the particular rice. Then uh, and economic like resistance, herbicide tolerant resistant, everything you should take care, like how much in economic, like for example you are doing hybrid, you want to develop hybrid idiotype, then what to take care, you should have a very amenable seed production technology like male sterility systems. You should take care of all the factors while developing any idiotype. Then, like what are the different steps we need to follow in idiotype breeding is first you have to develop a conceptual theoretical model. Like as I said, for a rice, developing a model, like you should have a semi-doff stature, then you should have the moderate number of tillers, erect leaves, like a V-shaped angle. Then you should have a very good grain number for panicle. Then like you should fix the number, how much grain number for panicle. Then what are the different weight of the panicle? Everything you are fixing before developing the breeding method. Next you are fixing, then you are started your breeding method. But in conventional breeding method, what will happen? You just first one trait, for example, yield or resistant, you decide one trait. You are doing a crossing. Well, in segregating population, you try to select the best individual, but not it is not in this case. Like in this case, it's a different scenario. Like first you deciding all the characters, like okay, this particular plant height should be this much height, the number of panicle this much number of panicle, number of leaves this much number of fields. Initially you decide the this character, then you are developing a one model, like one ideal plant type, then you proceed for the breeding. But in conventional breeding we first do crosses. It's like a trial and error method, conventional breeding, but it is target based approach, idiotype breeding. Then after deciding the model, then you have to select the material accordingly. If you want to develop cement of rice, you have to look for definitely the genotype which having these cement of genes. If you look up you want to develop a particular resistance, then you look for the material which having the resistance genes. Then after choosing all the material, then you start incorporating all the material into single genetic background. Then you start selecting the best plant and you do your breeding, like multiplication, testing, and try release. Then, which fact the factor which slows the idiotype breeding progress is symmetry and size of the plant pots. For example, like because it is also because of linkage. For example, you would want to develop some trait, but it is also linked with another trait. Means symmetry will be lost. Then size of the plant pot also. For example, deciding this thing is a very very difficult. For example, you are developing high weight panicle, but you want medium number of tillers, but something happen when you have a high like a high weightage panicle, then plant will droop, like it will drown. That's why like it will bend, but you don't want that. Like maintaining this symmetry, like synchrony in the, for example, while developing the idiotype, you should maintain the synchrony of flowering of each line, donor lines. Then the maintaining the plant's complete stature, symmetry of the complete stature, developing that is very difficult task. because we are bringing multiple donors, the genes into single limb. This is achieving symmetry and equal stature of the plant will be very, very difficult. Then compensation among the plant parts. Sorry, it's not traits actually. Compensation among the plant traits. For example, if you're increasing high grain number, then definitely there will be reduction in the grain size. Then how do you compensate it? That will be a very important task. Then pleiotropy. You're bringing one gene, maybe it having a dual function. For example, you are bringing a resistant gene, but it having a tip burning nature. Like for example, you are bringing a semi-dwarf nature, but it having a different color grains. Then how to do that? How to avoid that? If it is governed by independent genes, then you can do segregation and recombination. Then you select. But if the pleiotropy in nature, then how you will proceed that? That is one of the very, very important drawback of the idiotype breeding. Linkage drag and pleiotropy is very, very important points when you're reading about breeding breeding, like when you're developing an idiotype. Then genetic background. For example, if you are bringing some traits into a different genetic like a different genetic background, then definitely there will be a modifier genes. Means it will modify the expression of the particular gene. Then you will not get a desired expression of the particular gene that you brought from the donor plant. And these are the different factors like will slow down your idiotype breeding progress. Then the another aspect of
Sir,
yeah, yeah,
hello, sir.
yeah, ask her.
Sir, in case of pleiotrophy, if a desirable trait and an undesirable trait is controlled by a same gene, have they
Yeah,
attempted to separate those
it's very
previously
difficult.
ever,
Yeah, there is, we can do some attempts. You
like
can
using
put that
different
particular,
technology?
yeah, you can put like genes in a different backgrounds so that you can test, like for example, if it is gene is going for a resistance and also for the grain color you take, you put same resistant genes in a different backgrounds. Because of the modifier effect of the genes, the the expression of the particular trait may change. You may get resistant, but ideal grain color you may get because of the effect of the background genes, like the modifier genes of the background gene. That also that attempts done actually. Putting the pleiotropic genes into multiple backgrounds, then check in which background your background genes will overcome this effect, pleiotropic effect. Then you can choose that plant. Means you can compensating pleiotropic effect by the modifier genes of the genetic background that you are using. That attempts are done, but the any not this is the only attempt done. There is no any other modern technology to overcome the pleiotropy, because if the single nucleotide sequence may go in for two resistant genes, like two different character. No attempts are done, but several attempts are done, because in a single gene there will be a long sequence. And you know that now we reached up to single nucleotide level. It means if there is a change in a single nucleotide, there will be change in the trait. Then you can identify a single nucleotide by marker trait association, like by identifying marker trait association, by modifying that particular single nucleotide. May you can change that trait. That also done actually by CRISPR-Cas technology, genome editing technology. That has done actually, but not that much successful actually, because screening them it's very, very difficult. And first you identifying the genes which are responsible for the trait itself is a difficult. Like pleiotropic qtls are identified, but although which nucleotides are associated with particular trait, it's again a difficult task. Then validate, then you should create some mutations in that. Then you check for, okay, then there will be a change in the traits or not. Then you are getting a proper combination of the trait after screening of these mutants. Then you can do either only ultimate source is mutation that can be attempted, or you can bring these pleiotropic genes into different backgrounds. Then there will be effect of modifier genes on those pleiotropic effect. Then on which background you are getting good phenotype, like desirable combination of phenotype, then you select that background. Then you go for breeding. This can be done. Now you clear? Like your doubt is clear?
Yes, sir. Yes, sir. My doubt is.
Yeah. No,
Can
no,
we
no.
attempt, sir, this RNA interference like some technologies?
See, you can do anything. I'm telling that you should first identify which is the root cause for that. See, if you have a particular sequence, you can do anything. Either you can do any of the reverse genetic tools or any of the tools to modify that. First crux is, first you should identify which is the cause for this pleiotropy. Okay. What is the cause for the pleiotropy? If a single gene going for multiple regions
Single
means there will be
gene is
complex pathways involved. Okay. There is a complex pathways are involved in this development of the phenotype. At what nodes, for example, this particular gene having multiple traits, trait 1 and trait 2. Now we know that the gene can express through multiple genes, like the if it is end of the trait means first you need to identify gene. Okay, then this gene you are identifying, okay, it's going for two traits. If you identify the location and which, then if a gene is expressing means it is going through multiple pathways. There will be multiple nodes, internodes, there are hubs to develop this particular trait. At where you have to modify so that one you can eliminate the effect of the other trait, other negative effect of this particular gene. First, you need to identify. Once it is identified, you can use any method. Either you can use RNA interference, activation tagging, or expression, whatever it may be. Any tools you can use, CRISPR-Cas, or base editing, whatever, prime editing, whatever it can be used. But identifying this is a very, very difficult task because you should study the entire pathway of the particular gene at transcriptome level, metabolomic level. Okay, then like first you study the structural gene, like where it is located and what are the nucleotide which is responsible for this trait. Then you can study the gene at protein level, RNA level, and metabolite level. Then which pathway is responsible? Why this particular location having two phenotypes? After identifying that, we can modify. There is after identifying it will be easy. We have n number of techniques to modify this. Okay, now you understood?
yes, sir.
Now you understood or not? For, for getting why this gene is
Yes, sir.
pleiotropic is
Understood,
the
sir.
main point. Yeah. Then these are the different points regarding idiotype breeding. Then another concept nowadays many times asked in the exam, like last year they were asked what is designer crops. Okay, means you are modifying the genetics of the particular crop. You should have all desirable characters. That is only desired, like a designer crop. Then how you can do that? Like there is one technique called as breeding by design. Actually, this should be discussed after discussing all genomic approaches. But because it's linked to idiotype breeding, that's why I'm discussing today. What is it breeding by design? It's similar strategy like idiotype breeding. Like in idiotype breeding, what we will do? We are selecting the traits and we are trying to incorporate all the ideal traits into background, single background. Then we are predicting this particular background, like this particular developed idiotype, behave in a particular manner, like as predicted. But first you identifying the trait, then bring them, then evaluate. But nowadays, what we have, we can identify all the QTLs for this particular traits. Then you should study all the allelic variation of this particular trait. Then you develop all chromosomal introgressed as like CCSSL, right? So segment chromosome, like chromosomal segment introgressed lines. Then you just whenever you whatever the traits you want in the particular ideal plant, you based on the gene specific marker like anything, you just incorporate all that. Then you can develop the particular. Means it's like in silico breeding. Means you just design a chip like anything. Like one ideal chip, theoretical, like you having this particular allele here and you having particular allele in this here, then you can accumulate all the particular alleles combination in a single chip. Just ideal, I'm telling. Then this could be based on this reference, you can develop the lines. Means first we discuss in detail. First you map, then you develop introgression line libraries for all the loci for all the traits. Means you bring all the loci and all the low traits. Then you develop introgression libraries for each all the trait and all the allelic variation of the particular genes. Then you just check what is the effect of these allele on the phenotype. That can also be done. We can test the genomic estimated breeding values. That we will discuss while discussing genomic selection. Means we can now we are at the stage we can predict if there is a SNP, like if there is any gene or any allele, what is the effect of this allele on the phenotype that we can predict. Then we can bring all this the alleles together in silico, not in field. I'm not telling in silico. Then you can check when you have this combination of allele, what predictable phenotype you are going to get. When you have a different combination of alleles of all the traits and all the genes, then what phenotype you are going to get. Based on this, you initially decide in silico. Then you try to create the libraries among that. Then you identify, like then you go for instant, you construct one ideal genotype. Then based on that, you do your breeding. Then you can develop whatever the crop that you have designed in like first you design the crop, then after designing everything, then you start breeding for that. Okay, understood? First, I had to map the particular gene, then develop all introgressive library, then study this allelic effect on the phenotype. Based on the effect of allelic effect on the phenotype, we can check all the favorable alleles which are having good effect on the phenotype. Then in silico construction of this ideal genotype, then you can use it for breeding purpose. Okay, this is called as breeding by design. But in the case of idiotypic breeding, it's entirely based on the phenotype. After developing the idiotype, then you should predict that it should behave in a predictable manner. But here we know that which particular allele is having which effect on the phenotype. Then we can bring all them together. Then you develop in silico ideal genotype. Then you go for breeding. This is the difference between what is idiotypic breeding and breeding by design. Understood what is breeding by design? Many times, like last year in a survey, direct definition was asked. What is breeding by design? Means first you have to construct in silico construction of ideal genotype. Then you can use that in silico genotype for the further breeding purpose. Then last 2024 SRF also they asked what is designer crops. Okay, you can modify the genetics of the particular crop in such a way that you should predict in a you should behave in a predictable manner. Okay, this is called as the breeding by design. Then, what are the merits of these breeding by design and this one? We can make effective method of enhancing the yield by any morphological or physiological crop trait, for example, like higher photosynthetic ability, then bring that trait and you can improve that trait. Nowadays, conversion of C3 plant to C4 plants are taking place. It is also asked many times which genes are responsible for that. Means you can design an ideal phenotype. If you bring rice into C4, then what other component traits should include? Then how it will predict, like how it will behave in the if you bring the C4 system into plant. Then these are the things like nowadays this superized breeding also takes place. Means your super type phenotype concept to develop. Then based on that component traits, where breeding pathway is directed. But earlier it's a trial and error method. You just cross in a number of crosses, random number of crosses, then you identify better segregate, then we are going for breeding. But now because of the availability of genomic information or trait information, then you can use this ideal type concept and breed it. Like you can grain yield and direct and indirect product can be developed, and it is bridged between two gene pools. Then there are several hypotheses, like several advantages you can read. This is what idiotype breeding and the breeding design concept. Understood what is idiotype breeding and what is uh, breeding by design, and this concept was given by Donald in 1968. Okay, and you should remember what are the different ideal phenotypes was given in different crops, like cotton, who is given, like who given the concept, in brassica, who given the concept. Please remember all the scientist names which are developed the ideal type. It has regularly asked in the assembly examination. Okay, understood what is clonal selection, what is breeding by design, what is idiotype breeding, and what is multilines,
Sir,
and what is how
in
to develop different
sir, in
multilines.
idiotype and
Yeah. Hello.
in idiotype and in designer breeding, sir.
Yeah.
The only difference is we will deal in idiotype with only phenotype, sir. We only use the phenotype, but in this designer crops, we will use markers. We will deal with
Yeah,
alleles directly,
markers.
deal
Because you
with
have,
alleles. No,
yeah, that is the only great, and you can save time also. Then there you can avoid a linkage drag here, and you can study this. But pleiotropy is both the condition, and here you are developing the, you are dealing with that allelic level, so that you can change at any time. But here you can't study.
So
There
even
maybe, for example, you say,
transgenics can also be considered.
yeah,
For in designer crops, the transgenics can also be considered. So
can you like I'm not getting the point. Hello,
he
can you
is
repeat your?
asking, sir, can we consider transgenics as a designer crop, sir?
Yeah, that can be also done. Means transgenic means you are bringing a single gene, but you are developing all the traits and all the alleles of the particular trait. For example, if the one trait having the different alleles, for example, color you take, like you can first you can bring alleles of the all the colors, like red color allele, white color allele, black color allele, like this, like. But in transgenic, what we will do? Uh, there will be ideal phenotype will always one important line variety is there, but it is susceptible defect in one character, like in backcross, like you're bringing that particular gene from out of the gene pool, then you're developing a transgenic, like to recombinant DNA. But here we are doing, you can also develop the transgenic, like you can also develop designer for transgenic also. But here the difference is, see, we are dealing with entire plant like entire plant type, from germination to the yield. We are designing everything should be in the perfect order. Okay, you should have this much height only. You should have this much panicle length. Everything we are deciding. Then all the allelic variation we are tapping, which are the best allelic combination you are designing in silico. Then you go for development, because we are already having a libraries. So we are creating the libraries for each trait and all the alleles. For example, you take plant height. For plant type, all the alleles, one library. For another trait, all the alleles, another library. Means then which, for example, some plant you want this color and this height, then select this, pick the from the library the ideal allele, and this particular select this allele, then you do crossing and do develop. This is the thing. Okay. But this opportunity we can't get in idiotype breeding. In transgenic also you can do. First you develop the ideal type, then you introgress any trait. That also can. Okay? Your doubt is clear. Any doubts up to here?
He's asking charts, sir. Chart.
Yeah. So any integration library is similar to, yeah, it is similar to CCSL lines. Actually, it is CSSL lines only. That also can. These are like same as CSL, like CSSL line. Okay. Means every individual in CSL and what will happen? Like all the genome, when you take the entire library, it should having, it should represent the total donor genome. You are bringing the donor genome in the background of like recurrent parent in such a way that when you look the entire individual of the population, it represents the complete genome of your donor. It is similar to here. It is similar like CSSL lines. There the objective is different to map the particular gene. Here can objective is different, but it is the approach is similar. Through backcrossing, then we can bring all the donor genome segment in the background genome of recurrent parent, and when you take the all the individual, it represents complete genome of the donor gene. It's same one. Okay. Then any other doubts apart from this? No doubts?
I didn't understood this. That means how they are similar. Sir?
See, what is CSSL? Like a CSSL?
We take one donor
Yeah.
and we represent the donor genome in multiple lines, sir.
In a popular, one single background having here one donor gene. See, and not one, like some fragment of the donor genome. Then in the next, here again some fragment of the donor genome in the background. Then here some fragment of the genome. Then when you look at all the individuals of this CSSL population, it should represent the genome of the donor. Okay. Here also, each individual libraries,
Okay.
first you decide on plan, then you should create population libraries. Okay, like for example, here this particular, why we are going to develop this one? To study the, why we are going to develop this CSS lines? What is the main objective?
Gene function. We have to know function.
But you have to know. You have to know the gene function. Like if you are introducing in this particular background, then in this particular position, okay, at this, if you are introducing like this chromosome, the genes which present on this chromosome having effect on the phenotype. Here also you are doing. For what purpose you are doing integration line libraries? To study the effect of allele on the phenotype. When you are going to study, when you are introducing the alleles or a genes from a donor background to the known background at particular position, then only we can able to study the effect of an phenotype. Randomly, how can you say that okay, this is this allele having this phenotype, this is allele having this phenotype? How will you know? If you transfer to non-donor genome, this particular this, for example, here you are transferring in this line. If you there will be change in the phenotype in this line because you are transferring the donor genome here, there will be change in the phenotype. Then only you can know the allelic effect. That's why it is similar approach. Okay, maybe the aims are different, but the approach is similar. The idea is same.
Okay, sir.
Understood now? Okay, understood? Or
Yes, sir.
yeah. Okay, then it completes. I think idea type breeding and clonal breeding and multiline breeding. Then tomorrow we will discuss mutation breeding. Okay, then we'll discuss the system of varietal release and notification tomorrow. So I will complete. Then apomixis you read, I think it is similar. If I discuss also, if you read also, it's like beheading thing. I keep on reading the definitions. There is no concept here. But I can take one synthetic apomixis class, like after this, while discussing the CRISPR-Cas9, maybe I will discuss synthetic apomixis. Like the entire pathway has to be changed by baby boot gene. How we can synthesize the apomixis in plants, that can be discussed only after if you know the very like genomics. Okay, that's why we'll skip that to the genomics class, synthetic apomixis. But the basics of apomixis, types of apomixis, the different classification of apomixis, you should read by yourself. Okay, but the advanced part, like which are the different lines, how we can create synthetic apomixes, the how we can synthesize that pathway, that we will discuss after discussing genomics, like after discussing CRISPR-Cas9 and all those things. Okay. Good night. Good night, everyone. Any doubts you can ask?
Good night.
Yeah, good night. Bye bye.